Alright, Chapter 3 looks at cell structure and function, and we're going to look at a variety of different types of cells, and some key points to remember. Why are cells small? Things to think about as we're going through Chapter 3. Alright, what's different about human cells versus bacteria cells? How do things get in and out of cells? And some of these are just very good topics to think about, while we may not actually spend a whole lot of time on them in the lecture material itself. Alright, how do cells keep their shape and move? We humans do a lot of moving. We're constantly moving, even if we don't realize it. And how do cells get energy? Right, breaking down the food that we eat to get energy to supply our cells with fuel to do what they need. And then why do we need oxygen? Alright, so we talked a little bit about the types of theories that are within science, and one of them is cell theory. So which of the following are tenets of cell theory? All living things are made of cells. Most living things are made of cells. Cells are small. Cells are made by cell division. First cells were made in drying pools in Africa. Alright, so which one of those is correct? Well, the first one is the only completely true option based on the definition of cell theory. Option B, most living things are made of cells, is not correct because that most is not correct. For that to be fully correct, you would need to have all living things are made of cells. Option C is true that cells are small, but not officially part of cell theory. Cells are made by cell division. That one is borderline, maybe a trick option. Cell theory states that cells come from pre-existing cells. Doesn't necessarily indicate how the cells divide. And first cells were made in drying pools in Africa? Probably not, but who knows? Alright, what does the cell theory tell us? The material on the slide gives you the tenets of cell theory. As stated, cells are the basic unit of life and therefore are incredibly important to the organization of multicellular organisms. Each cell needs to function properly within its group or area for the benefit of that organism. As humans, we need our cardiac cells to support or to work together so the heart functions properly to be able to pump blood throughout our bodies. We need the cells in our pancreas to work properly so that all other cells are able to pull glucose out of the blood for energy purposes. Figure 2, or figure 3.2, is an important figure to think about. Why are cells so small? Why not just have a heart made of one huge cell? Alright, the reason is for cell surface area. Alright, with a lot of small cells, you have more surface area that is exposed to the environment. It is through the surface area that material such as glucose, oxygen, salt, potassium can be pulled into the cell for metabolic purposes. On the flip side, the cell is going through metabolic processes and producing waste material. If the waste is not removed from the cell, it will become toxic to the cell and the cell will die. If your heart is just one cell and it dies, then you die. An example of surface area and benefits is the following. A while ago, my husband and I were comparing the dietary information of a Chick-fil-A sandwich and the chicken nuggets. Looking at the protein value, those are pretty equal when you look at the 12-piece count chicken nugget. You had similar amounts of chicken. That made sense. But the fat content was much higher in the chicken nuggets. Why? Because the fat content is coming from the area that is fried. With a whole chicken breast, there is a smaller surface area that is exposed to the environment that can be fried. With the chicken nuggets, you have a greater surface area that is fried. Surface area for frying and fat content is not good for your health. Surface area for your cells is good because it allows movement of material across the cell membrane to be much more efficient at getting things in and out. So regardless of what type of cell one is looking at, it will need to be very small. Therefore, we need the aid of a microscope in order to see them. All right, the two major types of cells in living humans or in humans, you have the prokaryotic cells. We have a lot of bacteria in and on us. All right, prokaryotic cells, you see a picture of a bacteria cell here. All right, very simple. There aren't other parts to it. What you see is what you get here. They do have DNA, and then down here on the bottom you have eukaryotic cells. These cells have a nucleus, which you see right here. That's where the DNA is comprised, and there are different separate compartments, all right, such as mitochondria, these green guys, and endomembrane, right, all of this stuff around here, right? The eukaryotic cells have many similar functions between what type of cell you're looking at, and then when you compare the eukaryotic cell to the prokaryotic cells, you also have similar functions between the two different types of cells. All right, now, the plasma membrane is the two-layer of lipids that are going to surround the cell. It's the boundary between the inside and outside of the cell. It has a major role in maintaining the integrity of the cell. It is able to act as a gatekeeper for what goes in and what goes out. An example could be the security officer at the security gate at an airport. You cannot get past without a genuine ticket, proper ID, and a metal detection test. Therefore, not just anyone can get to an airplane. The plasma membrane is going to have a variety of different helpers to aid in this process. There's what's called the fluid mosaic model. That is often a term used to describe the plasma membrane. That is because each different cell type can have a different pattern made up of different proteins. All right, and those proteins are going to be embedded in the lipid bilayer, which you see here. Here's a protein, and you have the two layers of the lipids. All right, so those proteins are going to act as windows and linkage points. All right, linkage points would be places or material underneath plasma membrane in the cell itself. And then on the flip side, you can have different material that are going to bind to proteins that are found in the plasma membrane. So selective permeability means that the plasma membrane is able to regulate what goes in and what goes out. Selective permeability really means that some substances pass freely, some material need help getting through, and some materials just simply prohibited. So there are going to be three ways to enter the cell. One is going to be passive transport. All right, it's passive, it's not doing anything. That means substances move from high to low concentrations. No additional energy is required. Active transport, all right, it's active. So substances move from lower to higher concentration. It's working against the gradient, and you have additional energy that is required. It needs some active work going on, and that's going to be dependent on ATP or energy. All right, now looking at the plasma membrane in terms of why one might use saline to refill the blood, right? When you're given IV solutions, why are you not just given water? Right, water tries to get even amounts on inside and the outside of the cell. It tries to create an equilibrium. And you can have different scenarios that arise. All right, you can have an isotonic solution, which is what you find here. Isotonic, you have the same concentration of solutes, or let's say salt, on the inside of this red blood cell and in the water outside of it. So this would be saline. All right, it's an isotonic solution. the salt concentration is equal between the red blood cells and the liquid they're in. But if you put the cells in a hypotonic solution, the hypotonic solution shows that there's lower solute concentration in the liquid rather than the cell. What's going to happen is the concentration of the water outside the cell is greater, and so the water is going to move into the cell to try to maintain equilibrium so that the water and the salt concentration are equal. And what you can see from this picture, or sort of see, is that what happens is these cells swell. They get bigger because they're pulling water into the cell, which is what these arrows show. And in this picture, you can see the size difference. All right, and what happens then is these cells can get so big that they're going to erupt or lyse. Over here, you have a hypertonic solution, and so you have a higher salt concentration outside of the cell, and the water from the red blood cell is going to move out into the liquid to try to create an equilibrium. And so what happens to these cells? Well, they go through a process called crenation. And the cells shrink, they shrivel up, and all that you have left is really the plasma membrane of that cell. And so this is why one shouldn't drink high amounts of distilled water that you can buy from the store. It's why when you have IV fluids, you typically aren't given just plain water. You're given saline solution to create this situation and not this situation. And it's why when you are stuck out on a raft in the middle of the ocean, you can't drink the salt water, because this is what's going to happen. You're going to have cellular dehydration because all of the water is going to leave your cells because you have so much salt content. All right, and you can finish reading through the notes section on that. All right, so how do shells keep their shape? And so we're skipping a section here. Moving on to 3.5, there's 3 types of fibers composed of proteins that help the cells maintain its shape. All right, the 3 types of cells, or the 3 types of proteins, are going to be microtubules that you see here, these blue rods, actin, and then intermediate filaments. All right, there's microfilaments, intermediate filament here. You don't actually see the actin, but the actin is very important to maintain structure. Right? All of these molecules, these 3 molecules, differ in protein composition. They differ in location and the quantity as well, but they are all going to act to help maintain proper cell shape. All right, and this is where we're going to stop for part 1 of the audio portion and then move on into cellular energy.